tonight, we have two of our graduate scholarship recipients presenting. It's always some of our favorite lectures because we learn a lot, and we learn a lot about how what we're interested in as amateurs is applied on a professional level and goes very deep. So tonight we have two speakers. The first speaker is Shannon Meehan, and she is a second-year master's student at the University of Minnesota Duluth. And the title of her lecture is Effects of Disturbance History on Ectomycorrhizal Fungi and Tree Seedlings in Forestation. So Shannon's studying how ectomycorrhizal fungi communities can be restored in tandem with tree seedlings to enhance the success of reforestation projects. You can put questions in the chat for Shannon. Most of them we'll answer at the end of the presentation, unless something's really pertinent to what she's speaking about. Then I will lightly interrupt her and ask for an answer. So welcome, Shannon, and congratulations on winning the scholarship.
Thank you so much, Kathy, and thank you everyone for having me here. Yeah, thank you again. I'm Shannon Meehan. I use she, her pronouns, and I'm a master's student in Dr. Julie Ederson's lab at the University of Minnesota Duluth in the Integrated Biosciences program. And I'll be talking to you today about my research that's focusing on the role of mycorrhizal fungi in reforestation projects. And before I launch into my own research, I do just want to intentionally express deep gratitude for you and this incredible opportunity of your generous commitment to supporting research. 3 weeks after I received the exciting news of your award, I was then informed that a project that my research is tied to that's funded by a federal grant, that grant had been prematurely terminated. So just again, want to extend deep gratitude and let you all know that your support for the things that you care about really does matter, and I appreciate your action on that. And so before I launch into my specific research project, just want to give you a quick background about myself. I'm entering my 3rd year as a master's student this upcoming academic year. And before that, I received my Bachelor of Arts degree in Biology and Environmental Policy at Luther College in 2018, and then spent the next 5 years in a career pursuing ecological restoration and environmental education before returning to school. And it was these experiences in my professional career that helped me realize I really was interested in pairing environmental restoration with climate change solutions. And through that, I found the University of Minnesota Duluth and Dr. Julie Ederson, who is involved with a statewide project called Minnesota Million. And what this project aims to do is restore or reforest 1 million acres of Minnesota by 2045 with what we're calling climate smart tree seedlings. And when we say climate smart, what we're thinking about are tree seedlings from native species and genetic populations that we expect to do well as climate change continues to progress. And this is a really exciting opportunity for me because it's a collaborative effort across the entire state. So we have people who are going out and collecting seeds from tree populations that we expect to do well as climate change progresses. We then have local farmers growing out these seedlings and selling these tree seedlings to local restoration organizations who then plant those trees in areas that have been identified for reforestation. And all of this is not only just to reforest our areas, but prepare them for a changing climate and also work to sequester more amounts of carbon dioxide. And through this entire process, we're working to improve our research to figure out how we can improve planting success. And so it is that research planting success that my thesis is a part of. And I'll be talking to you about that today. So I'll give you a brief background about what I am studying and why I'm studying it. And then I'll talk to you about how I've set up my research. I'll dive into a few preliminary results. So as I've mentioned, I have 3 years or 1 more year to go this 3rd year. So I'm still collecting data, still analyzing a lot of data, but I do have some results for you that I can share today. And then I'll end with a brief conclusion before opening it up to questions and answers as well. So through all of this and through my research, I have an overarching guiding research question, which is asking, do natural and anthropogenic disturbances reduce the availability of ectomycorrhizal fungi to tree seedlings? And I'm just going to break down a few terms so that you all understand how I'm interpreting these words within my question. And so the first one, when I say anthropogenic, I'm just talking about something human caused. So in the bottom right of the screen, examples of anthropogenic disturbances are converting area to agriculture and or an orchard in this case. Or when we clear cut an area for timber purposes, that is also a human caused disturbance. And then when I'm talking about disturbances, I'm talking about any disruptive event at this point that has altered an ecosystem and caused mortality. And so these effects from those disruptive events would be apparent years after the initial disruption. And so that can be something natural, like a wildfire or a severe windstorm. It can be something human caused, and it can be abiotic or biotic. So abiotic is something like a wildfire or a windstorm, whereas biotic would be something like a disease outbreak that would maybe kill back a large population within an ecosystem. And a final term that I want to define just briefly at the moment is ectomycorrhizal fungi. So I'll dive into this in a little bit more depth in a few slides, but for now, just identifying it as a fungus that grows on the roots of plants, and it is a beneficial fungus that helps out plants, while plants also help out the existence of this fungus. And within my research, I'm focusing on the relationship between fungi and tree seedlings, because I know that fungi play an important role, as I'm sure you also know, in our forest ecosystems. And so this is a very general breakdown, but thinking about the decomposition of dead matter within our forested ecosystems is largely contributed to by fungi, and the fungi are restoring nutrients to the soil and making those nutrients more accessible across the whole ecosystem. We also know that fungi are an important food source for wildlife, particularly small mammals that then support other populations of other organisms within the ecosystem. And then finally, thinking about plant and soil health, and that's where my research plays in. I'm thinking about all the different fungi that connect the root systems of plants to the ecosystem, helping plants access more nutrients and water in the soil. And then fungi can also help protect plants from pathogens that may be in the soil as well. So with all of that, we understand that forests need fungi. And so when we're thinking about restoration projects and reforestation projects, I think we should also be thinking about what's been happening with the fungal communities, and are there ways that we need to restore fungal communities just like we do with plant communities, or are these fungal communities resilient and they're doing just fine on their own? And so with that, when I'm thinking about what's happening in the soil and the roots of all the plants and the fungi in this ecosystem, I'm specifically thinking about mycorrhizal fungi. And I love these photos because it just shows you how extensive the mycorrhizal community can get in such a short time period. So on the far left, we have a tree seedling that's been growing in soil with mycorrhizal fungi for only 2 weeks. And all those white cotton ball structures are the mycorrhizal fungi growing on the root system. So they found the root system of the tree really quickly. And then if we fast forward 3 more weeks, the photo in the middle is that tree seedling, and all those white fibers are the mycorrhizal fungi. So it's not the root system of the tree seedling, it's actually the fungus. And looking at an image like that, you can see not only does it improve the tree seedling's access to areas in the soil, but we're also seeing the fungus do a lot of work of holding the soil in place and keeping it stable within a system. And then the photo on the far right is just another image of a seedling that has a very small root system, and all those real fine white tendrils are the mycorrhizal fungi, helping that tree access more nutrients further out into the soil. And so when I talk about mycorrhizal fungi, there are 2 primary groups of mycorrhizal fungi that are probably studied the most. There's certainly more than just 2 groups of mycorrhizal fungi in the world. But the most commonly studied are arbuscular mycorrhizal fungi and ectomycorrhizal fungi. And so when I'm talking about arbuscular mycorrhizal fungi, I'm talking about fungi that grow inside the plant root cells. So they're actually infecting a root cell. And those dark blotches in the image on your top left, and really all of those images, those blotches are arbuscules or vesicles of fungi growing inside plant root cells. And we usually need to stain roots and look at them under a microscope to know if the fungus is present or not. Whereas ectomycorrhizal fungi, as its name implies, grows on the outside of plant roots. And this can be seen with the naked eye in some instances, but it's certainly easiest looking under a microscope as well. But you do not need to stain the roots in order to see the ectomycorrhizal fungi. And in addition to just how these fungi grow in relation with plants, they also associate with different kinds of plants in general. So if we think about prairies and the grasses and herbaceous flowers that are very common in prairies, those tend to associate primarily with what is arbuscular mycorrhizal fungi, whereas ectomycorrhizal fungi tend to associate primarily with trees, especially in forested ecosystems. And now when I say this, I recognize this is a very broad generalization, and both types of fungi can associate with many different groups of plants and across groups of plants. However, the trees I'm looking at and the forested systems I'm looking in do associate primarily with ectomycorrhizal fungi, so that is what I'll be talking to you about today. And so that wraps up my background section, and now I'll dive into how I've laid out my research project. And again, want to return to my guiding research question, which is asking, do natural and anthropogenic disturbances reduce the availability of ectomycorrhizal fungi to tree seedlings? And with this question, I have two hypotheses I'm testing. The first one being that disturbances that have severely altered the soil will cause the greatest reduction in the ability of ectomycorrhizal fungi to colonize tree seedling roots. So essentially, a more severe disturbance in an ecosystem will disrupt the mycorrhizal community. And then my second hypothesis is that tree seedlings that are inoculated with ectomycorrhizal fungi before we plant them out at field sites will have more fungal colonizations and experience greater above ground growth than their uninoculated counterpart seedlings. And so to test these two hypotheses, I have 5 field sites that represent real-world reforestation areas in northern Minnesota. And all of these sites exist on the contemporary and ancestral homelands of the Ojibwe and Dakota peoples. And I recognize that I am of colonial descent, and this acknowledgement does little to restore sovereignty to the original Indigenous caretakers from whom this land was stolen. But as I study the relationship between ecosystems and their disturbance histories, I want to recognize there are communities who have understood and cared for these ecosystems and their relationships long before Western science began to study them. And I'm grateful to be able to work on this land and understand what is happening through my research. And so as we zoom into these areas, you'll be able to see that my field sites all represent different disturbance histories. So up in the top left, I have a healthy forest system that is undisturbed and is serving as my control site. And then I also have a site that's the aftermath of a wildfire, a site that is the aftermath of a severe windstorm, a site that was forest converted to an apple orchard, and a site that was clear cut in the winter of 2023-2024 for logging purposes. And what I'm talking to you today about will just be forest sites. And so my wildfire site is not part of this study. It's part of a slightly different project. But the other 4 sites I will be discussing today. And so with that, I have 3 tree species that I'm working with. And my first species is the eastern white pine. My second is yellow birch. And my third is northern red oak. And I selected these 3 species because they're all species we're growing for Minnesota million. And they're also expected to do fairly well as climate change continues to progress, and they're native to Minnesota as well. And so once I selected my 3 tree species, I then, with the help of many other members here at UMD, planted 6,000 seeds, 2,000 seeds of each species, hoping that at least half of those seeds for each species would germinate. And this would allow me to have 100 trees per treatment per site, which I'll talk about in a little bit. And once my trees did germinate, I then moved on to inoculating these seedlings in the greenhouse. And so what that looked like is I'd go out to the mother tree stands where the seeds were collected from, and I'd dig up an amount of soil around the root system of these mother trees. And so the mycorrhizal fungi is growing on those roots that I've dug up. In addition to, there are also mycorrhizal fungal spores within the soil that I'm bringing back to the lab, and I add that to a blender with water, blend it up, and that becomes my mycorrhizal inoculant. And so we inoculated the red oak seedlings and the yellow birch seedlings, but unfortunately we did not have enough of the white pine seedlings germinate, so I only reserved those for one treatment, which is uninoculated and simply planted out at the different disturbance sites. So when I talk about treatments, I'll only be talking about red oak and yellow birch in the future. And once my trees were inoculated, I let them grow for a month before I then planted them out at field sites. And so when I'm transplanting my seedlings, I have both treatments. So what I'll refer to as a sterile treatment are simply the trees that were not inoculated before they got planted into their field site. And then I have trees in an inoculation treatment, which are the trees that got that soil slurry smoothie that should have the mycorrhizal fungi in them before they were planted out at these field sites. And this is a neat bird's eye view picture of what my field sites should look like. But the planting process looked something like this, a little chaotic, a lot of fun, and couldn't have done it without the help of many incredible members here at UMD and in the Ederson lab. So very thankful for all of that. And once my trees were in the ground, we then launched into data collection where we're measuring tree growth, particularly tree seedling height, stem diameter, counting how many leaves the trees are putting out. And then we're also measuring environmental parameters like canopy cover, how far seedlings are from a mature tree at their site, vegetation makeup, different soil parameters like soil nutrients, texture, soil density, things like that. And after the first growing season, which was last summer, I then dug up half of my trees to bring them back to the lab and look at their roots, where we can measure root growth, root length, and then count how many of the root tips are colonized by mycorrhizal fungi. And I'll also be sampling these roots for fungal DNA to see what fungal species are growing with my trees, if that differs between different sites, and if that differs from the inoculant that I initially gave the trees. And the funding that you all have awarded me that I'm extremely grateful for is being used to help me identify the fungal species through DNA analysis. So looking at mycorrhizal fungi on tree roots, it's very difficult to identify what species is there because fungus will look different based on what tree species it's growing on. And so DNA analysis is pretty necessary to identify these fungal species. And so that wraps up what my research is looking like, and I'll dive into some preliminary results for you all today at this point. And so first, I'm going to look at the colonization of root tips just by the disturbance. So based on what site these trees were growing in, how colonized by mycorrhizal fungi are they? And the first species that I have up here is eastern white pine. And on the y-axis, we have the percentage of root tips colonized by mycorrhizal fungi. And then on the x-axis, we have the different disturbances that the trees are growing in. And what we can see in this first graph is that trees in the orchard site had significantly fewer root colonizations than trees in the clear-cut site, but no other sites were significantly different. So there was only one difference based on disturbance with how colonized tree roots were here. And then if we look at yellow birch, it's a similar setup, but now it's different sites that are different. So we look at control, the healthy undisturbed forest actually had fewer percentage of root tips colonized than the windstorm disturbed site. And the other two sites were not significantly different from any other sites. And then finally, we look at northern red oak, and almost every site is different from each other here. So both the control and actually the clear cut were the most colonized by mycorrhizal fungi for northern red oak. And they were significantly more colonized than both the windstorm and the orchard. And trees at the windstorm site were also significantly more colonized than trees at the orchard site. So seeing this much significance for a species was pretty exciting to see. And then if I look at fungal colonization by treatment, so whether or not the trees received an inoculant before they got planted out, how colonized are they? And if we look at yellow birch, we see right away that there is a significant difference in that inoculated yellow birch trees had a higher percentage of their root tips colonized than trees that were never inoculated before they got planted out into the wild. However, when we look at northern red oak, it's not the same story. We actually did not see a difference in colonization regardless of treatment. And when thinking about that, so we've looked at disturbance and its impact on percent colonization, and then we'll look at treatment and its impact on percent colonization. But if we combine those two situations and thinking, okay, if a tree was inoculated and planted in a certain site, then how is the fungal community colonizing the root tips there? And this graph is a little bit messy, but I want you to focus on the square that's on the bottom left over sterile, and the square then over the inoculated column. And so what that is showing us is that the square represents my clear-cut site, and it is significantly higher in the inoculated treatment. So what that means is that trees that were yellow birches at our clear-cut site had significantly greater colonizations than trees at the clear-cut that were not inoculated. And that is the only significant difference across all of these sites. So all other sites, it didn't really matter where they were growing or whether or not they were inoculated. They had about the same percent colonization. And then if we look at red oak, I want you to focus on the triangles instead, which represent my windstorm site. And so if we follow the triangle above the sterile treatment and then go up to where the inoculated treatment is, that is also a significant difference and a similar trend as what we saw with yellow birch at the clear cut, where the inoculated seedlings in the windstorm are significantly more colonized by mycorrhizal fungi than the inoculated seedlings that were than the seedlings that were never inoculated at the windstorm site. But no other sites and treatments were significant for red oak. And then the next results I want to talk to you about are just the change in height of my seedlings across these different sites. So if we think again, just about disturbance, regardless of whether or not they were treated with a mycorrhizal inoculant prior to being planted, how well did these trees grow above ground? And with eastern white pine, there was no significant difference in their change in height. So on the y-axis, we have the change in height, and you'll notice that there is a negative value as well. And so we were observing what could be negative growth, but in reality, it was due to planting error, where some trees were planted deeper than when they had initially been grown in the greenhouse. So it appeared as if they shrunk over time, but that was not actually the case. And so across all sites, trees were not growing significantly more at any one site over the other. The same story for yellow birch, where again, we did not see much of a change in growth based on the disturbed area the tree was growing in. But when we look at red oak, we notice that trees at the clear cut grow significantly taller than trees at the orchard, but no other sites were significantly different from each other. And so that was just with disturbance. But if we think about the treatment the trees received instead, how is that affecting their growth? We can look at yellow birch first, and we don't see a significant difference. So regardless of whether trees were inoculated or not across all sites, the trees seemed to grow about the same amount over the first growing season. But then if we look at northern red oak, it's actually a different story where the oaks that were inoculated before they got planted grew significantly more than their seedling counterparts that were never inoculated before transplant. And then if we again combine, okay, if a treatment or if a seedling was treated with mycorrhizal fungi and then planted in a specific disturbed site, how does that influence the seedling's growth? And so with yellow birch, the biggest thing I want you to look at is, again, the triangle, so that windstorm site. And we see the triangle above the sterile treatment and follow it over to the inoculated treatment. And you can see that inoculated yellow birch at the windstorm site grew significantly more than their sterile counterparts or their seedlings that never received an inoculant before transplant. And none of my other sites have a significant difference from each other for yellow birch. And then if we look over at red oak, I now want you to focus first on the diamond that is above the sterile treatment and follow that up. It's kind of hiding behind the square, but that diamond is my orchard site. And in this instance, trees that were inoculated at the orchard grew significantly more than the red oaks at the that were never inoculated before they got planted at the orchard. And then something else that's really interesting to see here is if you look at the circle above the sterile treatment and then follow it over to the inoculated area, that circle is my control site. So it's my healthy, undisturbed forest. And the trees that were inoculated there actually grew less than and significantly less than the trees that were never inoculated. So seeing this relationship in an undisturbed location is really interesting to me and brings up more questions about what's going on between these different sites based on their disturbances. So those are all the results I have for you today, and I'll just wrap up with a brief conclusion where I'll return to my two hypotheses, the first one being that disturbances that alter an ecosystem will cause a reduction in the ability of ectomycorrhizal fungi to colonize tree seedling roots. And this is partially supported, but it depends on tree species. So we saw differences in, I believe it was yellow birch and white pine, but not red oak in this situation. And then in hypothesis 2, that tree seedlings that are inoculated with ectomycorrhizal fungi before they're transplanted at field sites will experience greater above ground growth than uninoculated seedlings and have more fungal colonizations. And again, this is partially supported and depends on the tree species and the sites that those species are planted in. So with all of that, the benefits of inoculating tree seedlings with mycorrhizal fungi likely depend on the disturbance history of a site and the tree species that you're working with. I certainly need to incorporate more environmental data in my models. So right now, I still need to account for canopy cover and soil nutrients and things like that. And from here, I'm excited to identify the fungal species within my treatments and also at my different sites to determine if it's specific mycorrhizal species that are impacting the growth of these seedlings, and if there are specific fungal species that are being impacted by the disturbances at these different sites. And so with that, just want to end with acknowledgements for everyone who's making this research possible, all the people who have collected the seeds for the trees that I've grown for this, the entire Ederson lab that's been supporting me through all of my research, and certainly Dr. Julie Ederson, who's been helping me and the rest of my committee design my project and analyze my data, all the sites that are hosting my research, and certainly everyone who is funding my research and making this science possible. And I also want to extend gratitude to the trees, fungi, and ecosystems I'm working with as I learn more about them. And so with that, again, I want to thank you all for your time and open it up for any questions or comments. And if anyone's interested in getting involved with Minnesota Million, you're welcome to visit climatesmarttrees.com. Thank you.
Awesome. Thank you so much. I do see a couple questions, and the one I think you've already answered, but Peter was wondering if you have any idea what species of fungi were involved in the inoculation. And I know you said that that's kind of the next thing you want to test.
Yeah, so right now I don't specifically know, but when I went back and said we were sampling root tips to see what fungal species are there, I also saved my soil inoculant or part of my soil inoculant, so that can get sent in with my root tips. And so what we see in the soil inoculant will tell us what fungal species were present in that.
Well, Aaron asks, were the tree types chosen for future commercial value in addition to their expected survival success?
Yeah, that's a great question. And I'm guessing the tree types is for Minnesota Million in general, and maybe not just my research project. But for my research project, I was thinking about trees I was somewhat familiar with. Yeah, what is native to Minnesota and expected to do well, and part of the Minnesota Million project. But yes, the Minnesota Million project is thinking not just about forested ecosystems, but also culturally important species, and species that could be important for other purposes like timber harvesting.
I was going to ask, and this isn't super scientific, but I know you talked about wanting to get a little more data on the environments that you're using, and I wondered if sometimes orchard sites, if they're using antifungal things, or if it's because there's sort of more of a monoculture there, that there's just less ectomycorrhizal fungi present, and then kind of the other side of that for the cut piece. It's like there were so many trees there and now they're all gone. Does it depend on what trees were there before? And are the fungi that are there kind of more hungry or more motivated because they just lost all their hosts?
Yeah, I think that is a fantastic question and certainly a scientific question, and would agree with all of those thoughts that you're sharing. And that the apple orchard, I don't know that it was ever like fully commercially productive. My understanding is they're more kind of feral apple trees that initially was found within a forest, and the forest was cut down, and then it was realized, oh, these aren't really edible varieties of apples. So it hasn't been treated with chemicals to my knowledge. However, it is much more of like a grassy area, and there's a lot of tansy out there, which is allelopathic. And so thinking about if tansy is releasing chemicals into the soil to kill things off, how is that impacting the fungal community? And I don't know what year that the forest was cleared for the orchard, but it's been, I think, a couple decades at least. Whereas with the clear cut, right, that was cleared winter 2023, 2024. And I think, yeah, before that was a very healthy forest system. And so there are papers out there that say mycorrhizal fungi can be resilient for a few years after a severe disturbance. But if they can't find a host to colonize, then that's when they start to dissipate from an area. And I would think similarly that, yeah, if the host isn't making sugars to share with the mycorrhizal fungi, that they probably are looking for other little living hosts. And so that could also be a really great hypothesis as to why I'm seeing what I am at the clear cut. So yeah, incredible question and so many things to explore for sure.
And then Randy's asking, did you observe mushroom fruit bodies of ectomycorrhizal fungi at the sites? And then it might offer a clue about what species are colonizing the tree roots.
Yeah, absolutely. So I will acknowledge that I am not a mycologist by profession. I would identify more as ecologist, which just means I'll know a little bit about a lot of things. But lucked out last year that we had a really wet spring and even into the summer. And so through that, I think the fungal community was really happy, especially out at my wind-thrown site. And there were tons of fungal fruiting bodies out there, and didn't know all of the species, but did try to take photos of things I was seeing and figure out, like, do we know if these are mycorrhizal or not? I was seeing coral fungi and I think wax caps, some chanterelles, and some boletes, I think. But yeah, just more than I know how to identify, which would have been really cool to have a foray out there, I suppose.
Awesome. That's super interesting. Any other questions for Shannon? Yeah. I found this super fascinating, and it's surely a ton of work. So yeah, thank you for yeah, we're proud to be a tiny piece of it. So thank you for sharing. Oh, John or Claudette, do you have any questions? Great. Awesome. Thanks again, Shannon. That was amazing.
Thank you all so much.
All right. I'm going to let Gregory share his screen while I introduce him. So our second speaker is Gregory Harris, and Gregory is a PhD candidate at the University of Minnesota Twin Cities. His topic of research is morticulture and the role of wood decaying fungi in snag dynamics. And we may learn more about this in his presentation, but what he had in his description is that morticulture is developing a set of forestry practices aimed at mitigating the loss of dead wood, both snags and down woody debris in managed forests. And then standing dead trees, commonly called snags, play an important role in forest ecosystem functions. Great. Awesome. Thanks, Gregory.
Certainly. All right, thank you. I've been really looking forward to giving this presentation, both as a member of MMS now for about a year and as a recipient of the MMS scholarship. I would not have guessed 2 years ago when I started my dissertation research in forestry that I'd be given a talk on mycology. And so this has been kind of an unexpected blessing and a passion that I've found. I've been fortunate both at the university here, some really strong mycology research, as well as the society being a very active and supportive organization. So it's been a real blessing. At the best, I was a mediocre morel hunter before all of this. And so let me start off. My first question was, why would a forester study mycology? And Shannon actually covered that quite well in her presentation. So I'll just recap that, for, there we go. For one reason, as she mentioned, it can be supportive to the plant's growth and survival, but it can also, certain fungi, or quite a bit of fungi, can be pathogenic to trees. Here's a picture of a red oak that was killed by oak blight up at Cedar Creek on one of my research sites. And the third reason is that fungi play an important role in nutrient cycling and decomposition. And that's the focus of my area. And specifically, I'm looking at snags. And snag is just a term for a dead standing tree. It was originally it was a nautical term referring to logs or trees in rivers that were hazardous to boats. Here's a picture from Scientific America in the early 1900s. They actually had snag boats to go up and down the Mississippi and other waterways to remove them. And it got introduced into forestry pretty early on. Here's a picture from the Flatfoot National Forest from a study in the early 1900s that mentioned snags. And I want to point out that there's kind of a pejorative term to snags. They've seen as a danger or obstruction, and that is played out in the literature for the last 100 years. Here are two articles. One by an entomologist here at the university back in 1925. He published in Ecology pointing out that basically deadwood is unique little ecosystems all in themselves. And but then on the other side here in 1929 in the Journal of Forestry, you see that another entomologist, Keene, was referring to them as, I've highlighted here, fire hazard, a menace, a burden, a serious problem. And just to drive home the point, he mentioned menace again. And it's true that snags, when it comes to forestry, can be a liability issue, a hazard for loggers or for people recreating in the forest. It could be a source of fuel for firewood and pathogens and things like that. So there is a concern about snags, but we're finding, or we've found over the years, that they're also a very important and critical component in forest ecosystems. You'll notice that Graham had mentioned felled tree trunk, and I'm talking about snags. So I wanted to just point out kind of a classification difference here. Dead wood in general is called coarse woody debris. And if it's upright and standing, standing dead wood, or more commonly snags. And if it's down as a log or large branch or even a stump, it's considered down woody debris. Much of the research kind of crosses back and forth between these two categories and what it's studying. I see that it's pretty important to distinguish the two and study them somewhat separately. You can see the snag on the left is a large white pine snag I came across up on the Chippewa National Forest, where a lot of my research is. And it provides a particular habitat for birds and other animals and fungi and all that. You can see the one on the right is kind of an entirely different picture. Why I like this picture, though, is that this is actually the top from that pine. When I went over there to measure the pine, I looked for the top, and you can see how drastic the decomposition rates are, or how significantly different that they are. And so it is important to consider snags and deadwood separately in research. So what do snags offer? Well, one is basically on the big heading of biodiversity. They can be structural habitat for both vertebrate and invertebrate species, substrate. Some of the organisms live within the dead wood almost their whole life, and it's certainly a food source. And when we say saprophytic species, we're really referring to any kind of organism that at some point in its life cycle depends on dead wood. So probably the picture on the left with the robin just roosting there temporarily could do that in a live tree and wouldn't be considered a saprophytic species itself, because its life cycle does not depend at some point on dead wood. But another big service that snags provide is being very involved in the forest carbon cycle. Just a little review on the carbon cycle. Essentially, carbon is taken up by plants, and here the trees, taking it up from the atmosphere, fixing it into organic molecules that's been used for itself. It can be released back into the atmosphere through respiration or through fire, and it eventually, a lot of it works down into the soil. In fact, almost 50% of the forest carbon is contained in the soil. Snags play kind of a critical intermediate from the above ground carbon to the below ground carbon there. And I'd like to explain that a little bit more, talking about wood in general and comparing forest ecosystem using economic terms. And when we talk economics, we think of currency and the flow of money. Where is money coming from, where it's going, and things like that. And it's really the same with when we look at ecosystems, we think of a currency, usually energy, and in this case, the currency is carbon. I mean, all life is carbon-based, and it's a source of energy. As you can find out when you burn wood in the stove for cooking or heat, it releases quite a bit of energy, and organisms can break down this wood and release that energy chemically and kind of in a slow manner for their own respiration and building and things like that. If you think about a tree is essentially like the income earner, it is getting the CO2 out of the atmosphere, fixing it, and then it is using it for its own respiration, growth, but it's storing a lot, almost like it's storing the carbon in a bank. And that bank is largely the wood. And you can see that wood, just the dry mass of wood, about 50% of that is carbon, give or take. And most of that is in the bole of the tree. The wood of the tree is composed of three components, largely. That is cellulose, lignin, and hemicellulose. Cellulose is the main structure. It gives its strength, tensile strength there. The lignin is more like a shellac that protects the cellulose there, and it is not as much of the component in wood itself, but it's very high in carbon, very concentrated in carbon itself. And it's a very tough chemical to crack. If you look at the rings here, these are aromatic rings. That is very difficult for things in nature to crack. This is very similar to petroleum waste that's difficult to break down as well. But fungi have cracked the code. Basically, the trees have died without a will, and there's all this carbon and energy up to grab, and the fungi have figured out through evolution using enzymatic and oxidative processes to break down this very recalcitrant, very tough to break carbon, get to it. And it's almost like then a stimulus money that's put into the economy. Once that is out and about, then it can use it, and other organisms rely on that as well. So I would not come to a MMS meeting without showing some slides of mushrooms, fungi that I've come across in the forest. All of these are Basidiomycota, and it's largely the Basidiomycota, or in that phylum, that the wood decay fungi exist. Many of these you've probably seen. Some of them you probably have eaten too. Some of them are edible. So why be concerned about snags in forests, and particularly managed forests? It's because managed forests tend to have fewer or lower quality snags. Here's a picture on the left of a pine plantation in just near Grand Rapids. It's about 75 years old. You see that the trees are fairly small. It's all red pine largely, and it's quite uniform. On the right, on the Chippewas and old growth forest, you see a lot more complexity there. There's several different age classes. The tree right in the center has been dated through dendrochronology. It's from about 1770. There's other red pines in there from about 1860, and then some from around 1900. There's also a mix of other species in there. So essentially, you have different species, different sizes, and when they become snags, then you have snags of different quality and sizes. So let's get into this term that I didn't even know about 2 years ago, and it's morticulture. It was coined by Mark Harmon, who studied deadwood for most of his career out at Oregon State University. He's now retired. He largely looked at down deadwood. He came up with he published a paper back in around 2001. And he was really addressing this paper to several groups. And he coined the term morticulture, and he made 3 points here. One was that he thought that this was addressed to the foresters. The first one, that we should move away from just the blanket removal of deadwood, because it's a critical component in forest ecosystems. And for a long time, the policy has been to try to get rid of deadwood in the forest, for the hazard fuel buildup, things like that, and liability issues. The second point that he made was that one should move away from what's called static minimum standards. This was largely addressed, I think, to the wildlife biologists, who have been very important in discussing the importance of deadwood in general. And what we mean by static minimum standards is that they would, in certain forests, identify animals, often woodpeckers, that relied on standing dead trees, and they would come up with these minimum standards, the number of dead trees maybe per acre that these woodpeckers or other birds needed in order to survive. His concern about that was, though, that they didn't see deadwood as dynamic. And I'll get into that in a minute, but he was saying that we should really move away from just thinking of it as a dead tree that doesn't do anything. And then finally, this is to bring in the culture part. He was proposing that deadwood can be cultured. And that's in the sense like agriculture or aquaculture or even silviculture that I'll get into in a second. And it's really when you move beyond the stage of just going out and foraging or hunting and relying on nature alone to provide something. If we want food in the future, we do agriculture. If we want fish in the future, we often engage in aquaculture and not just rely on what nature has to provide without doing anything. So he was proposing the same thing with dead wood. So what do I mean by snags are dynamic since they are dead and standing? Well, there's differences in their recruitment rate. Obviously, snags get recruited when live trees die, but trees die at different periods and at different rates throughout the stages of a forest development. And so you don't have the same supply of snags coming or getting recruited all along. And then once snags are recruited, there's differences in their longevity and in the decay trajectories. Here's a little bit of work or results from our work last year up on the Chippewa, where we were looking at the longevity of snags, particularly red pine. And we saw that in general, red pines can stand for about a decade once they die. You see a lot of clustering around there. But we also found, a bit surprisingly, that there were some that were 30, 40, almost 50 years old that had, died and been standing that long. This study over here also looked at some different rates of snag retention. We had basically historical data from repeated surveys on these forest sites. And starting off in 2014, we followed a cohort of snags, basically all the snags that were standing in 2014. And so over here on the left, one represents 100%. All of them were standing at that point. And then what happened between 2014 and 2015 was a series of treatments, forest treatments, basically different kind of harvests. And you notice from 2014 to 2015, the harvests themselves had very little effect on the retention of the snags. But a few years afterwards, it really started differentiating out. Right here is the 50% mark. So if you move over, this would represent when only 50% of the snags that started in 2014 were still standing. And you see quite some differences. I won't get into the details of the treatments here, but essentially they represent a gradient of disturbance level. The control being no disturbance at all, no management. And the transition treatment here was the most disturbing. There was regeneration cuts in there, some very heavy thinnings. And so you see the dynamics of the snags here. The other dynamic is just their decay trajectory here. This is kind of a simplified classification system of snags, how they change over time. Largely, they're losing height, their branches, things like that. But as you see with the next slide, oops, I jumped ahead, there we go, that there can be quite a variety in the decay and the morphology of snags here. The one on the upper left is actually that nearly 50-year-old snag that's on the Chippewa. It was about 7 inches in diameter when it died sometime between 1976 and 1980. It's still hanging out there. The next to it is kind of a common phenomenon I found up there, where this looked like a very precarious snag that was about to fall, but it's actually pretty stable with that core there. The next one you see probably had some advanced decay already in it, leaving that hole. So there's a lot of variety in these snags here. And when you start looking at them, you start trying to like, how do we make sense of all of this? And I think this is where forestry comes in to agree, because as foresters, we've had a good bit of practice on making sense of patterns in the forest. And I put this up here because when I was in high school telling people that I wanted to be a forester, I think these two images usually came to their mind. I'm quite sure because of the questions they asked me. They either saw the ranger on horseback and the ranger hat overlooking the forest, or logger. But there's so much more to forestry. And in particular, I want to discuss now a little bit about silviculture. And the best way to describe silviculture, I think, is when humans start interacting with the forest and wanting something from the forest. And the picture on the left here is from Cedar Creek. This is a closed oak forest, and down below, and this is just about a mile from it, they've been periodically burning the forest about every 3 years in this situation to create an oak savanna. Silviculture has been practiced for thousands of years. Really, it's the backbone of what we call forestry now, but it's been practiced. Indigenous people understood very well that if you wanted to create a landscape like the one below, that you needed to burn, and they didn't wait around for lightning to strike. They actually set fires to manage the forest and push it towards that state. As you can see, there's grass for grazing. Those are bur oaks and red oaks there, so you have mast for acorns and all like that. Likewise, on the right, this is on the Chippewa. This area here is a blowdown. It's a natural disturbance, but below that is a regeneration cut that was only about 1,500 feet from there. And you can see that there's quite some similarity in regenerating the forest. You saw that the blowdown created snags in the process, and in the one below, the tree there, while it's still alive, could eventually become a snag in itself. So when we think about silviculture, I like to think of leverage points, or where can we intervene some in the processes of the forest to direct it towards conditions that we might want. And then we can think about applying that to morticulture as well. Where are the leverage points in morticulture? For instance, one is in recruitment, and this is something that foresters already have a lot of knowledge and experience in. If you want a 14-inch red pine, say for poles, telephone poles, then, well, you can rely on nature and go find them, but you can also culture the forest and ensure that there's a supply of 14-inch red pines in the future. And so you see on this clear cut here, they've planted pines and also some natural regeneration. We get a 14-inch red pine. But the same concept can be applied to snags too. Instead of just looking for snags or hoping for the best, we can actually grow snags. And it's the same way. If you want a 14-inch red pine snag, then you need to grow a 14-inch red pine first. You see that they're actually quite similar. One is just a pole that hasn't been treated and left in the woods, and the other one's about to go up on the side of the highway. So recruitment is definitely, this is an area that we know in forestry, how to grow trees, and therefore how to grow trees that become snags in the future. So now getting into a little bit more of the mycology part, and the question that I'm looking at is what might be some leverage points to affect the longevity and the decay attributes of these snags. And One is relying on, I talked about the snags breaking down the wood, the cellulose, lignin, and all. As some of you know, they have different nutritional strategies. Some of them are brown rot. They have found a way to bypass the lignin. They don't really break it down. They're kind of like the kid that pushes the Brussels sprouts aside and goes straight for the mac and cheese. And so the brown rotters leave a high concentration of lignin from the what. And then there's white rotters. They consume it all. And they found ways to consume it all. And so how could these nutritional differences affect the snag longevity? Well, one idea is here. Juan is standing by a white pine snag that has brown rot in it. And brown rot is quite structurally much more unstable than white rot. I mean, they both lose their structural stability, but brown rot is that crumbly that almost turns to dust. So you had this large white pine whose base was getting brown rot, and it essentially buckled under its own weight and fell quite soon. The general thinking is that large trees, large snags, it take longer to decay. But in this case, because brown rot was in there and in the base of it, it fell much sooner. On the far right is a birch. You see the Ganoderma there, and that's white rot. It's still standing, but that is I was able to take my pocket knife and just plunge right in there, but it's still standing. There's still some structural support there. The top is broken out, probably through wind events and all, and it's just not buckled under itself. So if there's ways that we can influence the brown rot or white rot, which one is dominant in trees, that could be a leverage point there. And also, brown rot and white rot has a lot about the fate of the carbon. The brown rot is, like I say, what's left is the lignin. It's very recalcitrant, and that is more likely to get worked into the soil and stay for a long time in the soil, while the white rot is much more accessible by other organisms. In fact, there's some countries that use white rot as fodder for livestock. And so it's less likely to end up in the soil and get more incorporated into other organisms. So to wrap up here, we are going to start adding a mycology part to the study of snags here. We've done a little bit of preliminary work here on coring samples from dead trees. We're culturing them in the lab, and then we're using both genetic analysis and in the future hope to use chemical analysis to identify what species are in there, how they're competing, and also whether they're responding or whether there's brown rot or white rotters and to what degrees. And with that, I just want to conclude that horticulture is to me a very positive, forward-looking activity. As I mentioned before, there's kind of been an antagonism. You know, snags are bad, snags are good. I think horticulture is a situation where we don't have to look at trade-offs. We can kind of have our cake and eat it too. We can implement horticultural activities in our silvicultural activities to encourage the deadwood component, to keep it in there, and to actually improve upon it. Let's see. Oh, and I just going to conclude here. We see Neolentus there enjoying a cut stump. This is from a thinning there. It doesn't care that it was an artificial snag. Basically, I call it a mini snag. It is consuming the stump there. It's a brown rotter. It probably senses that that its supply is about up and it's fruiting to move on, but it's providing a very important component here in this forest of creating the calcitrant soil material that will get worked into the ground. Much more so than most people know this by the term train wrecker. So when it does leave the forest, it's kind of like a trip to Las Vegas. It gets a bit out of control, and it's known for in the past for decaying train trestles and causing serious train wrecks. So with that, let me stop sharing, and I'm happy to take any questions.
Awesome. Thank you so much. Just give me 1 second. I have you spotlighted. Gonna.
Okay.
There we go. Now, if people ask questions, you can see their face. And one of the first questions in the chat is, okay. Peter's wondering, have you reviewed the data on soil carbon storage of fungi? And Peter, if you want to unmute and ask anything more, clarify that.
Yeah, we've actually had presentations on some research, other research that was being done at the University of Minnesota, where they were calculating the amount of carbon stored in the soil from fungal sources, either live or dead.
Sure. That is particularly with emphasis on carbon, there's been a lot of work in carbon accounting, just, basically that I have looked at research there. My focus is more on the management of that instead of just accounting for. That's still a very important part, and there's a lot of good things to learn from that work. But mine is more as a forester, what activities can we do to basically either mitigate the loss of the dead wood or actually enhance it there. But yeah, there's a lot of work. At the last 20 years, there's been a lot of work in carbon accounting, and yeah.
And then Aaron mentioned in northern Minnesota, he's found that removal of lower tree quality in weedy trees had a greater impact on forest health and fire mitigation. For land that he has, that means removing jack pines and balsam firs. I don't know if you had any feedback on that, or Aaron, if you wanted to chat more about that.
Yeah, what was the... Let's see. I'm reading the question here.
Okay.
It's not really a question. It was more of a statement, or just more of a comment, really.
Oh, sure. Yeah.
Yeah, 3 acres, and that's what I'm doing is I'm removing all of the balsam firs and all of the jack pines and letting all the red and white pines and the birch survive, and black spruce.
Well, sure. I know one of my experiences last year, kind of systematically going through the Chippewa, that the jack pines were the ones that tended to have cavities in them for the wildlife much more readily than even the red pine, which was kind of our primary focus. I would say, foresters in the past have been really big on regulating the forest, maybe even simplifying it, that is, getting it down to one species. What we're finding now, and particularly with uncertain times with climate change and just not, knowing what the future will bring, it's good to keep as much as you can, all the components in a forest, or at least to keep your opportunities open there. So I generally discourage just eliminating a species. It's there for a reason, really.
Good to know.
And yeah, but I'm not saying. I mean, certainly, when we culture a forest, we might favor a certain species, but I think that the days are over where we just try to eliminate one, except for invasive species there. But anything that's natural.
And that was feedback. That was feedback from Cook County foresters, and so it's possible like their focus may have been more fire prevention, but I'm not really sure.
Well,
Without
that's true, and that is
there's
certainly an issue up there, and you know, Jack Pine
they
is
burn really well.
certainly yes, they do. They do very well. So yeah, again, it's a balance. And in that situation for you know uh, to prevent wildfires, it's certainly you could uh, maybe focus a little bit against the jack pine, but like I say, they serve a purpose as well. System.
That's really good feedback. Thank you.
Mm-hmm.
Yeah, and Shannon's asking that she's curious to learn more about how you age a snag, and can you speak more to this process? I was thinking that too. I'm like, how do you something so rotted?
Yeah. So actually, it's really difficult. We in a way cheated. We up on the Chippewa National Forest, there's a section called the Cutfoot Experimental Forest, and it's some of the longest term research that's been going on in forestry here in North America. And some of these studies have been they were started like in 1950, where they put in permanent plots, and they tagged or geolocated every tree, and they basically have tracked those trees over time. And now the study was about more timber harvest related or timber related, like thinnings and all. But the fact that they recorded whether a tree was live, had died standing, or fallen gave us some really good information there, and that is the history of those trees. And that's why when that one snag that I knew to be about 50 years old, it was recorded live in 1976 during a survey of that plot. And then the next time that they came through in 1980, they recorded it dead standing. So we know that it had died sometime in between that. It gives you a guesstimate, basically, but actually a pretty good one compared to that. There has been work in dendrochronology, which is generally used in live trees, but dead trees it becomes a lot more complicated because you are dealing with rotten wood and sometimes hollow trees, and so you don't get all the information to do that. There are researchers that have done that, and it's pretty intensive research there. We relied on these long-term studies that weren't even about dead trees themselves, but it gave us a history into those trees, and that's how we were able to age them.
Interesting. Shannon, did you have any other comments about that?
No, that was great. Very interesting to learn about. Thank you.
Perfect. And then Randy's asking, he says, I was intrigued with the photo of the brown rot next to the white rot in a single snag. Is that common? And are the dark lines between them called spalting?
Yes, that's them battling it out in there. That's a birch cookie from a study. Generally, that brown rot tends to be more dominant in conifers, and white rot in hardwoods. But that's not a hard and fast rule. And in fact, when you look at the birch, you see it occurring in both on that. And that's kind of part of where I'm looking at, and with the help of this scholarship, researching into that a little bit more. We're not cutting the dead snags down. A lot of these are permanent plots where we couldn't, plus that's dangerous. But the core sampling, we could sample around and take extractions there and try to get a picture of what's going on inside. And particularly, is white rot dominant, is brown rot dominant? And what some research is showing that it's actually not just a black and white or distinction. There's a gradient in there, that you have kind of gray areas where you have a mix of brown and white rot there. And so what we're looking at is, do some of our harvesting or some of our management activities, which creates different microclimates for the trees, does that influence the amount of brown versus white rot, or vice versa? And so that's something we'd be looking at in the future.
Interesting.
that.
I have a question kind of related to that, maybe, and it's when people are harvesting. I know, like when we're in the woods, we sometimes see something that has, been fallen, and you'll see a certain mushroom takes it over, and then another mushroom takes it over, and then when that's taken what it can, another mushroom takes it over. I think we've had some presentations that have kind of studied and alluded to that and showed us that. Is there a mushroom that's better when it's like, say you're harvesting something that rots something better or more safely, or would they ever kind of inoculate stumps to kind of be like, we want this, kind of degrading the forest versus something maybe that's pathogenic to nearby plots or something?
Sure, that's a really good question. It gets into kind of fungal succession, which is they call it substrate succession in there. If we think about the succession, say in a plant community, the substrate, the soil largely stays the same, and it's the composition that changes. What happens with decaying wood and in substrate succession is that who gets there first changes the chemistry and the makeup of that wood, which then could influence who gets there next, or who survives better there. That's why when you're looking at, say, the responses of, say, a red pine, it's under a strong genetic control. It can fluctuate in how it responds to the environment, but it pretty much stays in its lane that's set by its genes. But when we start dealing with dead trees, remember the tree is now dead. It's not responding to the environment. It's the fungi. And so you can take all these different routes of decay, largely depending on who gets there first. And they finding that in a lot of ways, there's some that are already there. They're just waiting for the tree to die. And it's the tree's defenses and largely its high moisture content that's just keeping them intact. Once it dies, it's on, right? That bank is there, and it's time to crack open the safe and get in there. And so if they're already endemic in the tree there, there's a head start there.
Interesting. Cool. Thank you.
Certainly.
Any other questions? I liked how your two talks kind of flowed and fed into one another. Sometimes you can't even plan those things. It was just
Right.
Here's the recipients who wants to go first, and it's really fun how this worked out. It was a really nice evening. Does anyone else have any questions for our graduate students? Let's see. There we go. Samuel, can you elaborate as to the agricultural value of snags?
I'm not sure if so. I was in trying to define morticulture, which is a bit of an obscure term, but I'm working on getting it out there. I was just drawing parallels with other culturing activities like agriculture and aquaculture, and then in forestry, silviculture. I did mention, and Shannon might be referring to that some I've read reports of some white rot wood being used as fodder for livestock. That might be, that could be kind of a silvo agricultural use there. I hope that explains. So there was a confusion with the terms that end in culture, or that was talking about the white rod that's sometimes used as feed.
All right. And then Randy's asking if you can comment on the ecological impacts of invasive golden oyster mushrooms in Minnesota. I don't know if you've seen that yet way up north, or if
I haven't personally seen it. I've heard of it. That's a bit out of my wheelhouse there. That's certainly like Bob Linchat's lab here at the university could address that. I think one thing we're finding is, though, that fungi are largely hidden and out of view. I mean, we see the mushrooms, but that's just the tip of the iceberg. And so and we think about invasive species, both plants and animals getting, through global trade and all, there's no telling what's getting passed around fungi. It's because it kind of comes under the radar there. And because again, they're difficult to study in the environment because of being largely microscopic most of their life there. So
It's funny, this is just anecdotal, and I know a lot of it has to do with, all the species in different areas. My aunt and uncle have some land, up in near Mora, and a lot of it's sandy up there, and I think it's a lot of poplar and birch and stuff. But he loves cutting trees, like he loves cutting trees, and they heat with wood. And I don't think there's a dead tree in his 40 acres. It's like as soon as like, oh, that's dead, I'm going to cut it down. And everyone's like, oh, you should go up there and go mushroom hunting. And his woods are too clean. It's like there's no dead wood. Also, it's like, on the other hand, my brother lives up north and he loves leaving the dead trees because of all the animal habitat it creates. And they like watching the wildlife. So it is interesting how those snags and the dead standing trees really have a niche for a lot of critters, and not just the fungal critters, but yeah.
But yeah, it's there's different. I know I looked at German forestry operations for a couple of weeks over Germany, and they think our forests are messy, and they're the type that they like to look like they practically sweat the forest over there. But part of it is that they're very into utilizing every little bit because it's been, more of a scarce resource in Europe, where there's been, use and wood use there that they very regulate it quite a bit, but then at the expense of things like the dead wood. The Scandinavian countries, Sweden and Finland, are doing a lot of work in dead wood there. I think they're starting to realize that it's an important component, and their intensive forest management, put fungi on the red list as endangered, and now they're doing a lot of work in that area to address that and mitigate the loss.
I mean, I think especially in go ahead, Claudette.
I was going to say, even with all the ashes that are dying, I've noticed in the neighborhoods, just in Burnsville, they'll have a tree service come out and they'll trim off the outer branches of the tree and take it, and they're leaving the main trunk standing. A gentleman behind me did that with 2 of his trees. Another one up the street also did that. And around the corner again, people are leaving the bigger chunks of the trees. Now, I don't know if that's economically because they don't want to pay that amount of money to have it all taken down and gone, or if they're looking at that this snag or standing dead tree is providing habitat for a variety of birds.
Well, I think it's both. I'm glad you brought that up because that's a kind of in the future a venture of mine, and that's deadwood in urban settings. There's a tree just behind Borlaug Hall here where they've done that. They limbed it and they cut it down to a point where it's safe. I mean, obviously, hazards in urban areas, it's more of a concern, but it was intentionally left to be a snag. And I also met an arborist, had dinner with him and a couple people that he's had people request that, and he's doing that now. And I think that's a great thing. I mean, people shouldn't be freaked out by a dead tree. There, you can plant flowers around it, and you're going to get, wildlife and other benefits there. And also just disposal. I mean, the main volume is in the base of the tree there. So that's one less thing that you have to haul off to wherever they dispose of it, which is a cost. Why not leave it there and still provide a function and service?
Right.
Did you guys see on the news? It was just this last weekend, and I don't know if it was on like Lake Harriet or Lake Nokomis. Anyway, someone had a large, very tall, straight tree that had died. And I know some people do like a chainsaw carving on or whatever, but they had turned it into a giant pencil. And every year they do like the sharpening of the pencil. And they said like there were 2,000 people there this year, and they made sort of like an event out of it, but it truly was a standing dead tree, and they just made it into art. And now it's sort of a spectacle. I like that idea too.
Yeah, I thought that was great too. They covered it on Care 11.
Yeah. It's like, I mean, it doesn't really, it kind of, I don't think it's a habitat for anything anymore because it's all painted up and stuff, but I don't know. It's an impressive.
Well, I'm sure there's fungi. Great. I'm sure there's fungi in there.
I'm sure there is. Yeah.
They're masters at taking advantage of
things
there. But no, it's good to see more of that in urban settings and just getting away from what I call cemetery landscaping. And you'll see the bee pollinator and the nomome and things like that. And I think leaving some snags, if they're cut down to a safe, level and monitored, that could be a real benefit.
We have a large stump and kind of the part of the trunk left of it that it was just too large to cut by us and take away, and it's sort of slowly rotting away. And this stump now, I mean, it's nearly gone, but it probably was a 200-year-old oak. So it's probably 4 feet across, whatever, this stump. And every year it grows a chicken of the woods and a hen of the woods. Yeah. And my husband's, and I'm just like, my perfect stump that I can rely on for just a little edible and not have to go anywhere. And my husband's like, I could park another car here. It's time to get rid of that. And I'm like, you can get rid of it when it's fallen apart and doesn't grow a hen. But until then, and so that I've taken a poll of other mushroomers. I'm like, would you remove it or would you keep it? And everyone else says keep it. So I still win the argument for the time being.
That's good. Yeah, I know at the Basilica of St. Mary, I'm a member there. There's 2 elms that they had been spraying and trying to keep alive, and they finally succumbed. And I started trying to hint this whole leave it as a snag there. But again, they like that kind of lawn, or what I call the cemetery look. So it may take a special order from the Pope to keep it, get it as a snag. But it would be nice. I mean, here's a tree that's lived for up to 2 centuries and could still provide a service in the area.
That is totally fascinating. We've got an abandoned farmhouse in Taylor's Falls, and it has junipers planted out front of it, which I always called cedars. And someone had come by who was I don't know if they're forester or botanist or what, but they were interested, and they said, these are not indigenous trees here. These were planted. They were most likely planted as a part of the landscaping for the house when it was built. And we know the house is 1868. So he's like, based on the size of that, I'm going to guess it's probably from, within 10 years of that, or within about the time of the house being built. And it's hard I wouldn't know that. And I like to kind of learn that and kind of think about something is, like you said, some of those trees have been there since the 1700s. And it's harder for us to think past our own kind of like human life scale.
Yeah, I know in forestry, we start thinking a little long term, not as long term as geologists, but certainly, beyond the quarter or the fiscal year, you really have to think ahead.
Perfect. Awesome. Well, this was a great discussion. And one more note from Randy that he has a white pine stump that grows train wreckers every year. Those are fun. All right. Well, if there's no more questions, I want to thank you guys so much for being here tonight.